Aloha, and this is a very kind of a little bit of a off-putting first metagenomic DNA honey. Whoa! Forage and invasive species. Hopefully, it's not going to be that heavy duty when we get into it. But this is what we're doing, and metagenomic will explain. But as Franny put it, wait, you detected LFA, DNA, and honey? I did not even think that would be possible. What a wild tool. And that's kind of what we're going into is that this is developed into something we didn't even know we could get. And it started with my husband and I questioning, well, what's in our honey? And then developed into a STIRS project. Can eDNA, which is environmental DNA analysis, provide baseline botanical source pathogenetic identification for citizen science beekeepers with year-round forage? This is a USDA NIFA STIRS grant that we got. And it started in 2019 with curiosity. As Franny said, we started beekeeping in 2016. So after 3 years, we started learning a little bit more. And we noticed every single time we took honey, especially month by month, completely different honey. Obviously, we knew it was from the different forage, but living in a tropical rainforest with literally hundreds to thousands of plants, it was like, what are they foraging? And that middle one, that real light yellow one, a local beekeeper said, "Oh, you have Ohia honey." We are in the 1900s. Throughout the whole 1900s, our land was cane field, and 17,000 acres around our land was cane field, and didn't change until the 1990s. So about 30 years of change. So there aren't any huge old growth ohia forests. There's not a lot to say, okay, we've had ohia. So that got me even more, where are these bees going and why is there so much variation? Now we do know that bees are foraging and they forage flowers for pollen and nectar. Pollen creates all the fatty acids, all the amino acids, all the what we think of as heavy duty nutritional stuff, all the vitamins and all that. Nectar, which we always think of, is purely sugar. It's the energy that they're getting. And bees take nectar, which is sap, which is almost all sucrose sugar, and the flower starts inverting. And we always think of, ooh, dangerous inverted sugars. Actually, honey is an inverted sugar. Inversion means that it splits that sucrose into fructose and glucose. So this flower actually starts it, and bees go and using an enzyme they have in their honey crop, they invert using invertase enzyme into honey, and that makes it a thicker solution, but they also dry it by passing that honey back and forth. And the big thing that bees are looking at is making that bee bread, that thing at the bottom. Bee bread is a high nutrition fermented product where they use the sugars from from their nectar and the yeasts and the amino acids and fatty acids from the pollen to make this highly fermented, easily digested food that they feed to their baby bees. So a lot of it has to do with baby bees. Now, there are two things that aren't on this. These are what we always think of them foraging. They forage two other things: water, which they use for cooling, and also just in feeding, making sure that the honey that they use is the proper consistency. They also forage tree resins for their propolis, which they glue everything together, but it's also a very good antimicrobial substance. We're not covering that because we didn't actually study that directly. And there are nutritional differences, and in Israeli studies, which is another year-round type forage area, they did notice that there is complete nutritional variations, not only year-round, but also geographically. And the whole bees are looking at balancing their protein that they have and all the little micronutrients and their amino acids and their alpha 3s and alpha 6s and carbs, make sure their brood, their little baby bees, are the healthiest they can be. Now, we, when we started this, we're like, well, how can we get these answers? And everyone was like, Melissa, pollinology, looking at the pollinology of honey, Melissa. And we don't think of honey having pollen in it, but it does have some pollen grains. And the old-fashioned way that's been used for hundreds of years is you wash off all the honey. Literally, you wash it off, centrifuge, and then you get a little fine dust. And in that dust are pollen grains. And looking under a microscope, you identify them, which seems kind of easy if you have a few tens of pollens to look at, different pollens. When you're talking about hundreds to thousands, it becomes much more complicated. And plus the fact, with our ever increasing number of different species of flowers here, you get ever increasing number of pollens to know. And a typical person who's a specialist in pollinology has about 20 years under their belt before they start doing good identification. I didn't have that long. So I also found out that the newer method is using DNA extraction and then using PCR, which many of us heard thanks to COVID. We all had PCR tests to see, do we have COVID? Much of us did not realize that PCR is actually breaking up the polymerase chain using a polymerase chain reaction and looking at individual little tiny snippets of DNA. And then you want to get an exact match. And then if you're looking at just trying to identify, you look for those exact matches in the amplicons, the things that you're making. And in PCR, you're making multiple copies of that same little tiny snippet. And you try to find out, can we identify it using databases? But you're only using a little snippet of a portion of DNA, and you're amplifying it. And in that, you can have some errors coming in. There's some intrinsic errors that come into that. And that's what we were looking at doing with our grant, using multiple primers, multiple PCRs, but then a newer, much more dynamic potential came up right as soon as we got our grant monies, and that was metagenomic sequencing using shotgun sequencing, where you take all of the DNA material, fragment it, and then all of that DNA material, not amplified, not in any way changed, is put through the DNA analysis. And then you're looking for the DNA. Now, a lot of people know, oh yeah, ATCC, I've heard of those. And there are four main deoxynucleobases, Thymine and adenine are always together. They're double bonded. And guanine and cytosine are always together. They're triple bonded. But cytosine has a way of breaking down, and it can break down to xanthine and uracil. But those are all triple bonded. So you'll mainly see A, T, C, and G in combinations. And they can go, you can actually get a TAAT, TATA, and those are different than a ATAT, TATA. So those are really important to know. And these are actually the protein, the amino acids that make up all of life. So this code is literally called the barcode of life, using four main nucleobases, these acids, to make up all of life. We kind of don't even think that that's possible, but it's when you take hundreds of thousands to millions of these pairs, base pairs, you can start getting differences in each form of life. Now, how does that DNA get into the honey? Well, you take a nice fuzzy body, which everybody knows honeybees are nice and fluffy and fuzzy. And then as they fly, their wings are beating against their body, causing static electricity. Anybody who's ever played that balloon on their head, you rub your hair and your balloon becomes a nice static magnet. Well, that's what the bee is. But then you add a little bit of stickiness to it, and it really goes well for collecting things. And they can even collect when they're flying in the air, but especially when they land on that flower. Anything on that flower will be drawn to it because the flower itself usually has a negative charge, and these bees are positive charged. And as they clean themselves, they're even more positively charging, so they're antenna. Everything is sticky, and then it can get into the honey. Once they go back to their hive, they're going to start cleaning themselves. They clean off their antenna from anything, and they're walking all over their hive. Well, the hive has honeycomb wax, sticky, and in those little honeycomb cells are honey. So little bits can fall off as they're grooming and walking, but they also can transfer it between bee to bee because bees transfer their honey back and forth, and that's called trophallaxis. They do that to increase that invertase, that inverting sugar, because they want to split up that sucrose as much as they can. They really like high fructose sugar, but they also like high glucose sugar. It's not as usable, though, for them. And they're transferring back and forth all that honey. And in that, you can transfer everything they've gotten on their bodies. Now, this is some pictures of bees collecting that I've taken from our yard. And they're collecting all sorts of different things, including ornamental flowers, nectars, but you can especially see in that center one, that lilikoi, that one bee in the center is drinking the nectar. The nectary on that flower is right in the center. And many people say, oh, bees can't get to the pollen because those are up in the pads put back. But they walk all over this flower, and that flower has lots of little speckles of pollen. If you look real closely, you can see those little speckles, and the bee below it has lots of speckles. So it has pollen, it has plant material, and anything that visited that flower beforehand. If a big old carpenter bee came by and was on there bubbling about, some of its DNA might have gone on that little bee, especially the bee on the bottom as it drops down. But they collect, and then that lower right hand is trophallaxis, where they're actually two bees are sharing a drop of honey, and they're sharing it back and forth and getting it nice and thick and what we like to think of as honey. Now, shotgun metagenomics is a study of multiple genomes from multiple organisms recovered directly from an environmental or biological sample. And we're using a biological sample, and that is honey. And the sample reads, oops, I want two, I didn't want to go two. Okay, the honey, the first thing, you got to remove the honey, and it seems odd, but the honey is actually masking that DNA. And you just have to wash the honey out, spin it in a centrifuge until a couple of times until you have a nice fine powder at the bottom of your tube, and then the DNA is extracted from the residue. Now they fragment everything and then go and sequence, and they fragment it using both mechanical and chemical. Normally in PCR, they'll use one or the other, but this way they normally do both, fragmenting chemically and mechanically, and then they're sequenced into reads, and then they try to repurpose putting that puzzle back together, seeing where those DNA codes cross over and to make a complete read again. It seems backwards to break it up and then make it go whole again, but because of the way DNA sequencers are, they only have short reads, about 300 to 500 base pairs is all they can do. And remember, DNA can have hundreds to millions of base pairs. So they're matching them back up, and then they're compared to worldwide base databases. Now, some people, especially in our Hawaiian samples, you're going to see a lot of unknown genome. And they go, well, why are there unknowns for errors? One of the things could be there's low biomass. There's only one or two particles of that DNA. It would be harder for it to identify, especially when they try to put that puzzle back together. There could be environmental contamination, and we did see that when my husband and I first did our first sampling. We weren't sure if we had a grass that we weren't sure of, because bees don't normally go into grass, And my husband had also done some weed whacking before he went into the hives. So was it weed whacking error or was it an actual read? It turned out it was an actual read, but it was one of those things we didn't know. And then it could be that in remaking, when they're reading those strings, rather than being nice long skinny strings of DNA, they may have looped and created a little bit of a different read. If it loops over, it's going to read it without that loop. And those are called chimeras and stuff like that. And they're also, and more likely in Hawaii, incomplete reference databases. There's very little in the world gene banks on our Hawaii specific plants. And especially when we're bringing in plants, animals from every direction, almost all temperate and tropical areas. We're bringing those plants in. So we're talking about tropical rainforests around the world. We're talking about temperate areas around the world. Now, the reading DNA is kind of like using an like identifying with a key, if you've ever done that. And if you might have a small leaf sample and look at a key, you'll know that you can't really get a good read with just one little bit. And then you might have some bark. And does anybody yet know what this is? If you know when these two things, just raise your hand or do a reaction. Okay, now you might have a bit of a flower, but it might have a insect in it, so that kind of makes it harder to read if you know what that is yet. You might have seeds. Great way of knowing. Anybody know what this is? Now, many of you are going to know what it is now. Those of you in Sacramento and other places around the world might have a harder time because this is ohia. Is ohia polymorpha? Metricidius polymorpha, and that polymorpha is a really important thing. That means it has many forms, and some of you might go back and go, that's not the ohia leaf. I know it's got a little oval, real oval leaf with lots of fuzzies. And yes, some of them do have inflorescence, and some of them do have pubescence and stuff like that, but some don't have. Some don't have the inflorescence with the yellow tips. Every ohia pretty much has a uniqueness to it, almost every. And that's why having World Gene Bank data from our specific, especially when we have valley to valley differences, is really important to get. Okay, now in this study, in the SIR study, we took honey samples from 22 apiaries, and they're the yellow dots high on the east side and a few on the west side. And people go, why didn't you do as many on the west side? And that's, we are asking people, it's citizen scientists, so we're asking beekeepers to come forward. So if any of you are beekeepers interested in sampling, we'd love to have your samples. And these were taken from last September through last December, and it's a mix of targeted samples taken in December and random samples returned throughout the fall. And they were sent for analysis at Salvia CCAS in Estonia. And that's a big thing because they are the first ones to do this metagenomic shotgun for honey. And we are actually establishing the Hawaii database for them. But they have worldwide sampling of honeys, and so we went with them. And we got results, both FASTQ files, which are now at Bishop Museum and at UH Manoa in the Pacific Cooperative Studies Unit, and also at UH Hilo at Ilila Biodiversity Lab. And we also, every single beekeeper got their 2 interactive corona displays, which are really cool files, and also a pathogen parasite chart based on worldwide known honeybee pathogens and parasites. And these charts, the one on the left is all species, and that includes the one on the right, which is just the plant species. So you can play with just the plants if you want, or you can play with everything that includes the plants. And these charts are just crazy playing with because you can search by the species, and yes, we're in Latin base, so you got to do the scientific names and all that. It's kind of cool because you can do little tiny searches. But the reads of concern, the chart on the left is the one that they sent us, that Sylvia sent us, and then I just went with all of them. The highlighted yellow ones are the ones that came up in the honey samples that we have. And the main ones everyone had were small hive beetle, greater wax moth, and one of the spiroplasmas, um, which is also called May disease. It's normally found throughout most areas in spring only, but these were taken in fall. We don't have high incidence of varroa, which is pretty much known because by nature now we kind of have resistant um, bees to varroa, and we don't have high incidence of nosema or the lesser wax moth. Now, on the left, there's a huge chart that I've done because one of my things was, okay, what about toxic honey plants? If we have toxic honey, we'd want to know some of the toxicity is toxic to bees, others are toxic to human. And most of what we have is toxic to bees. We do have castor bean tree in 6 of the samples, which can cause human illness, and we also have euphorbia, which can induce vomiting. But the big one, that pink red one, is one that is really high concerning because in New Zealand, where they have this plant, this one's from Nepal, but it's the same toxin and it has the same reaction. In New Zealand, they actually have to test all honey during their flowering season because of the high toxicity. It's highly poisonous. It has caused human death. There was only one sample and low counts. These are low read counts, so we're not as concerned yet, but it's something to look at. It's something to definitely check. Is this a real read, for one? And then if it is a real read, is it a one sample, one plant sample, or is it wider spread? Now, going into this, when I first found out about Teuton plants, I wasn't really concerned because you have to have 2 species for it to be a toxic honey, because bees do not normally utilize this plant. They only utilize the honeydew that a moth called the passion vine hopper, its caterpillar, when it's eating away, it drips down honeydew, and bees collect honeydew. And that honeydew is toxic. It's not toxic to the bees, so they're able to transfer it to the honey, but it's toxic to humans. And so without having a moth, you don't have to worry about it. So I wasn't as worried, but I just did a search in Hawaii to see if we do have the moth, because I didn't think we did, because it is an Australia, New Zealand only moth, and it was noted in 2022 in Oahu. And it's in the invasive species. It's also in the Department of Ag. There's an agricultural because it is a hazard to lilikoi production. So it's something we should be looking at. Now, other reads of interest. We found ag pests. We found 2 different types of thrips. We found even nematodes, which really the Achilles typale was found in a sample that I wouldn't have thought you'd find a nematode in DNA that a bee would get, but there are many ways it could have gotten it in water sampling it, or if it landed on soil. But we have different species of fruit fly, the Drosophila, and also even aphids. And then known invasive insects, we have our favorite, everybody's favorite little fire ant, and also the Argentine army ants. So we have 2 of the known invasive species ants, But we also found some endangered. We have the Hylaeus anthracynus, which was found in 3 samples, and they were not necessarily known to be one over in the Kona side and two in Upper Hilo area. So that's something that some of the people who are into Hylaeus study are looking at. And then we have some novel reeds, things that haven't been seen in Hawaii. We have two chimatinas, and those are little tiny green insects. We have some wasps, really cool. The Teleopsis has its eyeballs on the end of those antenna-looking things, so it's kind of a cool ant-looking thing. But we have a number of insects we didn't know we had. No record of it. Is it a real record? We don't know. This is stuff we have to check doing ground truthing. That's not part of our grant. We're hoping to build on this grant for obvious reasons. We also have not noted in Hawaii apis, the bee species. And the big ones that I'm looking at are the Hebrota laberosa and also the bombus. Bombus is bumblebees, and a lot of people say, I know I've seen a bumblebee, but it's never been recorded in Hawaii, and 10 samples had bombus, had bumblebees. So do we have bumblebees on Hawaii Island? There's a pretty good chance there are. Now, I'm not as worried about the apis species because some of those could just be, again, in making the puzzles, the puzzle pieces fit, but they weren't really good fits. We probably, there is a good chance we do have Apis cerana, which is it looks like the honeybee, but it has some different ways of dealing with things, but that is something to look at. Now, the concerning thing about bees as a whole in Hawaii is Hawaii's had one of the highest biosecurity on paper starting in 1908. We did not allow any living honeybee or materials to come in because Hawaii was so afraid of getting a disease that had spread across the US, and that was American foul brood. We did get it by 1930, which obviously meant we didn't have as good a biosecurity in action as we did on paper. And we've gotten every disease that bees can get from the United States over, but there's usually a 1 to 2 decade buffer that we have before we get it. So that means our biosecurity works, but not as good as we could have. But it is not legal to bring any used equipment unless it's certified through ag department, and it's got to be highly sterilized. And no living bees are allowed in. Obviously, for research, there are some, and only honeybee semen is allowed. So if we do have apis species, that could mean people are going around those high strict standards for our biosecurity. Oh, this gets really good. These are all the known invasive species reeds and insect reeds. I just put this up. Everything that's blue, we showed that we had hits on in the honey of bees. So it's kind of cool that you can actually do some monitoring. And if there was anything that was of major concern, we know the location of the hives that they were found in, and somebody could actually do some ground truthing to see if it is a high concern. The most common plant reeds, which is what everybody wants. What's the most common? We've got to have all this great stuff. Is Eucalyptus grandis. And this was done in, again, the fall, September through December. Now, I've got to really stress on this one. It was the fall of one of our longest drought seasons. And people go, well, what does that matter? Well, eucalyptus is normally found in that upper orange #1 area of that map. So that salmon orange area. We don't have high below that, and yet almost all the reeds below also had high eucalyptus. And remember I said they can pick up samples while they're flying in the air. Eucalyptus is a known Hawaii air pollen for people with asthma and stuff. So it could have been that during a drought year, we had a lot of air pollen. I haven't found yet the records for last fall. Love to find them, but we don't do as much on Hawaii Island because we have so much rainfall that air pollen is not normally a concern. But I'd love to know if anybody did readings of that, because that could be one of the methods. Those little sticky static bodies flying through the air could have picked up airborne pollen, which can travel tens to hundreds of miles. But for especially town locations and lower Puna where eucalyptus isn't grown as heavily, it does seem a little bit off. But in Puna, citrus, orange and cane grass and paperbark, which everybody has always seen, were noted, and also this Polygonodon cinnamon, which is a novel species. And if you've seen a plant with a flower like this, let us know, because it's a novel one. It's not noted yet in Hawaii. So
Hey, Carey, sometimes your audio is feeding out.
okay,
Can you say that again?
yeah.
Okay.
Okay. In Punas, there's orange, cane grass, paper bark, and also this polygon cinnamon, which is not noted to be in Hawaii. So if anybody has in Puna, if anybody sees a flower like this, let us know, because that's one of the ones that's a novel read we're not sure of, but it did come up as a top read in some of them. And then in Hilo, along with eucalyptus, also myrtle trees came up, and Hamakua, along with uh, eucalyptus, top was also an ash tree. And then in Kona, they did not have high eucalyptus. They had Hiave, Lilikoi, date palm were the top counts. So a lot of people go, "Whoa, I wouldn't have expected that," but that's what we're getting from our samples. Now, when we look at this is from one of the Kona samples, and this is what, if you open up a chart, this is what you're seeing. This is the chart, and you'll notice there's at the top there's a big search bar. You can search for anything you want. I mean, I've had some kids look at it and they go for some weird little things, but you don't even have to do the full name. You can just start. But I clicked on Viviplanta is all living plants, and you'll notice that there are 117,278 segments, sequences of plants, and it's 10% of the whole of that chart. And that high grade thing is that other, that unassigned. And we'd love to know what 24% was, but we don't. But these are, if you wanted to know, well, what all can you get? You can get bacteria, viruses, plants, um, fungus, animals, and then a whole bunch of different others, in the microbial world. So you can get a lot of different information from this chart. And we're going to just open up plants. And I'm going for some things. We're going to go into Gentianalis, to find out, because we're over in the Kona coffee belt. We're going to look for Kona coffee. So I've opened up the plants, and this is what the plants in the second step in plants. And then we have coffee. And if you look up at the search results, I actually had just done COFF in search to get there, and then I backtracked to get the pretty ones. But we have Coffea arabica in there, and Coffea arabica there were 19 sequences, which is about 0.02% of the whole plant species. So not a lot you don't need a lot to actually find it. And to find coffee in these were September samplings. So to find coffee within September, you're not talking about many blooms. So it could be that they landed on a coffee plant because blooming time is normally spring. There could be some rogue blooms, or it could be they just landed on a coffee plant and got some coffee DNA with them. And now I'm going on that same sample and I'm checking out funguses because this is to see, okay, can we get both sides? Can we get the plant and then agriculturally significant things? And opening up the funguses, we're going through, reading through, and now that bottom thing that's highlighted, that little blue spear, which has hardly anything, it has only, here, I'm going to open it. Oh, wait, nope, I want to go backwards. Okay. Okay, that bottom spear, that Buenavera bassiana is actually put on coffee plants now to counteract coffee borer beetle. And we had one sample out of all 22 that actually had shown that that fungus is actually on the plant. They're actually treating it for coffee borer beetle, which is kind of cool. But then if you look over, that whole green area is all fusarium, which is another not so great uh, plant to have, fungus to have on your plants. Okay, and I went through and did all of the coffee funguses, and we only had coffee shown because it was not bloom time in 5 out of the 22 samples. 3 of the samples were Kona coffee belt, so the bees have a lot of coffee to land on, just to land on. And then 2 of the samples were in the Hamakua, the cloud forest area where they have cloud forest um, coffee. And in all of our samples, we only had that one that from Honolulu that had the B. bassiano, the beneficial. But we did have a number that had ones that are pathogenic funguses, and we were just looking at coffee, but some of these are pathogenic over other plants also. And one of the Kona samples had 3 of the not so great things, including Cercospora and um, Fusarium coffeteum, which is directly against, it's a Fusarium that's directly against coffee. So it's interesting to note, we didn't know we could use this tool for that type of thing, but we can actually look at honey and find out agricultural beneficial and pathogenic funguses that are available. Okay, so the conclusion, metagenomic DNA has more data than past studies. It's a way of identifying forage species and forage species in all types of forage, not just plants alone. And you can ID plants along with pathogenic and beneficial microbes, and also animals. And these chronometagenomics that comes from Salvia, they supply them with the results, provides interactive data display. It provides search features, and it's very easy to use. You don't have to have a lot of DNA expertise to use it. Now, what we need now is some more research, and this is not funded by our grant because we were just seeing, can it be used as baseline? Now that we know that we have some baseline data, we want to look at possibly growing this into verification with ground truthing, longer term studies, and also a lot of people want nutritional and chemical data on these. And if anybody wants to do anything, right now we're in the pre-proposal, Dr. Jonathan Koch at Manoa Pacific Studies Cooperative Unit is doing a survey. So if anybody can, you can click on this beautiful little QR code for that survey. We would love it if you could fill it out. It takes about 5 minutes. And it's to find out what is Hawaii, of the people in Hawaii, and even those outside of Hawaii, what research results would you like to see if we do extend this to a larger study? And then if you'd like to do my survey for Sarah's always wants to know what the participants have felt about this presentation. So if you want to do that, that's this QR code. So get your camera up, click that, and do that. This is full of QR codes at the conclusion. Okay, and also for people who want to know more about PCR, Radiolab, which is presented through the National Public Radio, had a great little episode called The Age of Aquaticus. Just look at that or copy this YouTube, and it's a great explanation of PCR and just how cool a tool it is. And then if you want to play with the charts, we have 3 charts up. Hopefully by the end of this week, as I said, this is 22 charts are up right now on these, and they go from left to right. There's September, October, and December. And I know November's missing because that's out in the 40 that are out coming back hopefully this week. But we will have from September all the way into April in this next series. So, and if you look at the written part, you just really have to take one of these QR codes because the last 5 digits are all that are going to be changing, and that's the 3 date, the September, October. The next one will be NOV 25, and then 26 will be the ending, and then Jan, Feb, March, April, that type of stuff. Okay,